So the next RFI presenter is Aaron Hingorani, and the title of his talk is International 100,000 Consortium Drug Development Resource. And Aaron is from the University College London.
Thank you. Thank you to the organisers for pulling together this second summit, and thank you for the opportunity to present this proposal. So the proposal is to focus some of the activities of the consortium on drug development, specifically to develop resources that link genotype to clinical phenotype and molecular phenotype at scale to enable drug target identification and validation. And I'm presenting the proposal on behalf of colleagues in London, Usha Menon from the UK Longitudinal Women's Cohort and Juan Pablo Casas in Boston. But I know there are a large number of individuals in the audience and elsewhere who are interested in these types of efforts. So this is really a platform for wider discussion and to scope interest in taking something like this forward. So the summary of the research idea is that the consortium is in a prime position to support drug development and repurposing because of its ability to link genomic variation to biomedical data at exceptional scale. And that this huge resource has the potential to de-risk drug development by reducing the currently high rate of drug development failure. And the hope is that this would enable academia and industry to more often work on the correct therapeutic targets for each disease, and to enable work on new mechanisms of action and diseases that have previously been considered a bit too risky to tackle because of poor insight on the underlying mechanisms. And the translational aim, really, of this proposal is to focus drug development on those diseases where there is unmet clinical need to the benefit of patients and societies globally. So I'll say something about the motivation and some of the evidentiary basis of the proposal. So the major motivating problem is the very high rate of drug development failure. Perhaps as many as 96% of drug development programs fail. And that has consequences that affect us all. If we take healthcare systems and patients, what we find is that those successful drugs that are developed, prices are often pitched at a level to try and recruit the cost of prior R&D failure. And that in turn puts financial pressures on healthcare systems, and in publicly funded healthcare systems like the NHS in the United Kingdom, where I come from, with universal coverage, there are then downstream consequences on governments who have to provide healthcare. They have to raise taxation, divert funds from other sectors, or limit access to less cost-effective drugs. For the pharmaceutical industry that's engaged in the endeavor of trying to develop new medicines, the very high rate of failure can instill risk-averse research and development, and therefore move away from developing new novel compounds, risky compounds with new mechanisms of action. And when failure occurs late in clinical development, the costs can sometimes be high, particularly for pivotal drug development programs, and that can have knock-on consequences on research and development, contraction in workforce, and so on. So the idea is that actually we all have a stake in good drug development if we want to develop new medicines for a wider range of diseases. So the question is, why should failure be so common? And I think there are several ways of thinking about this, but two of the main processes in drug development are, first of all, identifying the proteins that are most important in each disease, because most drugs currently act on proteins, whether they're small molecule drugs or monoclonal antibody or peptide therapeutics. So it's this protein target disease matching problem that lies at the root of much drug development failure. Of course, once the right proteins are identified, the second step is identifying compounds that are specific for that protein target. So more about this target disease matching problem. I think I've frozen. Can anyone help? Slide won't advance. Thank you. So why this problem of matching the right drug target to the right disease? Well, the traditional approach to trying to identify which proteins are important in which diseases has been to study preclinical models, isolated cells, tissues, or animal models. And these turn out to be an unreliable predictor of treatment efficacy in humans. Moreover, there's only a capacity to treat a few targets at a time, or to test a few targets at a time in these preclinical models. And there's an increasing concern in biomedical science, particularly wet lab, preclinical biomedical science, that there's a very high false discovery rate, a lack of reproducibility of studies. And what that might do then is result in a number of flawed target disease hypotheses being taken forward into clinical phase testing. And of course, the definitive experiment as to whether a drug works, acting on a new protein works in a disease, is the randomized controlled trial, which is the final step in drug development. And if you fail at that point, there's been a huge wasted, effectively, investment. So can we do any better? So the idea is that genomics may help in this respect. And the principle really stems from this idea, this Mendelian randomization paradigm that's emerged, which is that natural genetic variation in a gene encoding a potential drug target, whether it be in regulatory sequence or encoding sequence, that affects the expression or activity of the encoded protein, can provide insight into the effect of a drug acting on the same protein, and moreover may even provide insight on mechanism of action. The advantage of a genomic approach is, of course, that we're studying the correct organism, the human being. With genome-wide approaches or whole genome sequencing approaches, there's the potential to study all potential drug targets in the genome at the same time. There's no target selection step involved. Researchers in this field over the last decade or two have taken great pains to try and reduce the false discovery rate in genetic analyses. And of course, the randomized allocation of alleles at conception according to Mendel's laws emulates the randomized controlled trial design. So effectively, genetic studies are like natural randomized controlled trials, and there's a potential to study all targets across the genome in every disease every time. So you might think of the issue here, or the problem, being a large number of diseases and a large number of potential targets. And you might think in the bottom right-hand corner of the slide as a kind of matrix of diseases as columns and genes as rows. So if there are 10,000 diseases, as there might be at a certain level of ICD-10 coding, and there are 20,000 protein coding genes, there are potentially 200 million unique protein disease pairings. And the task in drug development is to identify which of those protein disease pairings is causal. And so expanding one section of that square at the bottom, the top left section, and blowing it up a little bit so you still have diseases as columns and genes as rows. When you do a genome-wide study, you're really interrogating natural genetic variation across all genes in the genome in that disease, and you might generate some hits. Then the second step that you're interested in is, do any of those hits map to genes that encode potentially druggable targets? So those are the little orange squares as opposed to just the blue squares, which are causal only. So this approach has some evidence. So genome-wide association studies reviewed over the last decade or two have already rediscovered the known targets of drugs to treat the diseases for which those GWASs were done. Effectively, this is a positive control data set. And we estimated in 2015 that the genome-wide association studies done to date had rediscovered, either through their indication or mechanism-based adverse effects, about 80 drug targets from the 600 or so licensed drug targets that were accessed at that time. Moreover, we've heard a little bit earlier on about phenome-wide association studies. By knitting together genome-wide association studies, or specifically focusing on a particular gene identified as a potential target in a GWAS study, and using variants in the gene as a proxy for drug effects, and testing the effects of those variants against a wider range of diseases, can provide information on target validation. Is there likely to be an adverse effect of modifying this target on another disease? Or is there another disease that might also be beneficially targeted by a drug developed against this particular protein? Moreover, if one of those drugs is already in clinical use, but for a different indication, that opens up a repurposing opportunity. So there are already emerging examples of this. This is just one. So variants in the gene encoding the interleukin-6 receptor were identified in a genome-wide association study of rheumatoid arthritis, and a drug, tocilizumab, a monoclonal antibody, is already licensed for the treatment of rheumatoid arthritis. But the same variant has been identified in genome-wide and other genetic studies of a range of other diseases: coronary heart disease, atrial fibrillation, abdominal aneurysm, and also recently inflammatory bowel disease. So there's a potential here to think about this target not just for rheumatoid arthritis, but for a wider range of inflammatory and related conditions. We heard a little bit earlier about this particular finding from Peter Wirtz and colleagues from Finland, showing that these phenome-wide association type studies can be extended to deeper insight on molecular biomarkers. So this study compared the effect on the circulating metabolome measured using the Nightingale platform that we heard about earlier. So looked at the effect of a naturally occurring variant in the gene encoding the target of statin drugs on these metabolites, and compared the effect of taking a statin on the same metabolites. And you can see that the profile on the metabolome of taking a statin drug was reliably anticipated by variants in the gene encoding the statin target, albeit a much smaller effect, so the data have been scaled up for the genetic aspect. But you can do a thought experiment, which is that if you didn't know the effects of a statin on the metabolome, but you had the genetic information, that would provide you insight into what a clean drug targeting that protein would do. So what about the proposal? So the idea would be to develop a genomic data-driven drug target identification and validation platform where there's a standardized agreed approach to data generation, meta-analysis, visualization, reporting, and sharing in a form that's useful for drug development programs, whether they be in academia or industry. Since many of the operational tasks of an initiative like this are similar to those that have already been very successfully deployed in genome-wide association studies for disease gene discovery, and many of the cohorts within the consortium have already participated in these, I think it would be reasonable to expect that useful new knowledge for drug development could emerge within a 2 to 3 year time frame. In terms of the activities needed to achieve the goal, in fact, many of the enabling activities were described in the previous session and actually could be deployed to achieve this sort of translational aim, to give those enabling activities a little bit of specific translational focus in a way. So one element is to scope the potential for different cohorts to be able to participate in an initiative like this. Let's call it a coordination strand. The importance of harmonizing clinical and molecular phenotypes across the participating studies. We heard a lot about that in the previous session. Wrangling existing genomic, genotype and sequencing data into a form that allows cross-cohort analysis. A lot of this has already been developed methodologically for genome-wide association and similar studies. We've heard a little bit and talked a little bit about the potential to enrich participating studies. For example, studies that already have rich clinical phenotype data, but whereas yet their DNA has not yet been extracted or genotyping has not been done. And in other cohorts, perhaps to consider enrichment of molecular phenotypes, proteome and metabolome, using some of the platforms that we've heard about before. It would also be important to agree the type of analysis which will be done. We heard a little bit about the idea of code to data versus data to code, so an analytic strand. And there are a number of methodological issues that I think would need to be grappled with, but there's a broad range of expertise across the cohorts and collaborators that I think could be brought to bear on some of the issues that affect this type of work. There are some additional policy issues and challenges. We've heard a little bit about information governance and what data sharing models would need to be put in place for this sort of genotype to phenotype linkage to be done. And we've heard a lot about federated meta-analysis type approaches being ones that actually fulfill many of the scientific needs, but yet fulfill the information governance and data sharing requirements of many of the cohorts. Since I think there'll be a lot of potential industry interest in this activity, models also need to be considered that recognize the contribution of citizens' genomic and healthcare data to the value chain. So there's an additional level of policy issue here that I think the consortium would need to think about this academic industry collaboration that might emerge from this type of activity. We were asked to say why cohorts are needed for this type of work. So I don't think I need to rehearse this again, because we've heard a lot about why cohorts are well placed to do this. I think cohorts are well placed to do this. I think rich case collections are also needed too for certain diseases. And Adam Butterworth talked a little bit about where there are some diseases where we don't really fully understand the environmental and genetic basis and have poor treatments, and we can make some inroads there. But I think the key features here are of scale, the breadth of phenotypes, and deep resolution, including molecular phenotypes that can be brought together with a consortium of this size. So I've specifically shied away from being very specific about the type of resources that might be required. It really depends on the scale of the initiative and whether new genotyping, new omics measures, and so on are in scope. And I think a much more detailed costing would be required if the initiative is prioritized by the IHCC. And I think that needs much wider discussion. It could take the form of a very large strategic initiative, for example. So in summary, the proposal is to use genomic and linked biomedical data at scale for drug target identification, validation, and repurposing to reduce the high rate of drug development failure. The idea is to help academia and industry to work on the correct therapeutic targets for each disease and to try and stimulate work on new mechanisms of action and diseases that have previously been considered too risky to tackle from a commercial standpoint. And the scope of funding, the model for collaboration, and potential industry partnership would need wider discussion. Thank you.
Time for questions.
Meg M. from GlaxoSmithKline. I'm curious if there has been any discussion around the availability of some of the samples to be able to do target validation work that this type of work would definitely lead to. Because certainly I think the opportunity to collaborate with any of the cohorts to actually perform the target validation studies would be a very important aspect.
Thanks. It's an important question, and I think different cohorts, from what I can see from the table of cohorts, are at a kind of different stage of maturity in converting samples to genomic and other data. And I don't think it's a question that I can answer for all consortium members, but I think it's an important one. But I think many cohorts will be in a position that have such samples available, will have the right governance arrangements and so on, and may be ready to move.
I think in the portal that you're talking about, the openness or the available, to those types of collaborations would certainly be an important factor that I would want to consider in terms of this work.
Thank you.
So in terms of using genomics for target validation, there was an effort 5 or 6 years ago or so, a fairly broad one. Lon and I were involved in, and some others that tried to engage industry in this, and my understanding is it didn't really catch fire. And I wonder what has changed since then that would make it catch fire now.
So I wasn't involved in that, so I'm not sure what extinguished the flame last time round. So I don't know, maybe others who were involved may be better placed. I don't think I could answer that well. But I think we have strong evidence here, and I think both in academia and industry there's a recognition that this type of approach may have a role to play. So maybe the timing's right now and it wasn't then.
Well, and it may have been that there wasn't the infrastructure built that we're
Right.
talking about building here. I don't know if anybody in the field, thinks about how it was 5 or 6 years ago and how it is now. If you have any comments on that.
Yeah, there is actually recent consortium work where this has sort of succeeded.
Yeah.
And looks like even in eMERGE it's happening. So.
I'm sure, Arun, your charm would bring everyone together in a new way. Yeah, so it's long been known that most of the drug targets are associated with traits that aren't the ones for which they're indicated, right? That is a truism, I think, amongst all of these. So I guess my question to you, apart from getting the industry partners involved, and I don't think that's a safe assumption to assume they'll all come running for various reasons,
Yeah.
for legal and other reasons. If you did this and you had a big catalog of all the targets for existing therapies and their associated diseases, isn't that just a recipe for off-label prescription?
Not necessarily. I mean, how you use a medicine, a particular medicine may still need to be validated through a trial for a new indication. Payers may require that. And if it got to that stage, then the license may be modified as a consequence. So I don't think it's inevitable. It's not necessarily a bad thing if we did in certain cases. I mean, we prescribe off-label as clinicians frequently. Most medicines in children are not licensed for the indications they're used for. They're extrapolated from adults. But I think my motivation as a clinician is to get a wider range of treatments for a wider range of diseases.
So, Terry, I do want to comment on something. Some thoughts about. I wasn't involved in that earlier effort, but I think it's really important to realize that that the work that you propose there talks about identifying genes. That is not the same as identifying a drug target, and I would say that what we're finding is actually identifying the gene is I don't want to say simpler, but is a shorter time period than actually turning that gene into a target. And so I think that if you were going to develop this to something that industry would be interested in, then you need to engage the industry about how you go from the gene to the target.
Completely agree. I don't think it's the finish, it's the start, as you say.
I was going to point out actually what is the difference between that and
But I think it's the right start point, and things will fall by the wayside. We'd expect them to, but hopefully less frequently than they have previously.
Yeah. Well, thank you, Aaron.
Thank you.
And the next speaker is in the RFA series is Daniel MacArthur from Harvard, affiliated with Broad, the Mass General, and he will speak on the human knockout project. Daniel.